Welcome to this video about oxidase test. In this video, we'll explore what oxidase test is, how it works, and how it can be used to detect disease. We'll also discuss some of the advantages of oxidase test and the current state of the field. Let's get started. Before we dive into the topic, let's figure out the meaning of oxidase. In biochemistry, an oxidase is an enzyme that catalyzes oxidation reduction reactions, especially one involving dioxygen, O2, as the electron acceptor. An important example is cytochrome C oxidase, the key enzyme that allows the body to employ oxygen in the generation of energy and the final component of the electron transfer chain. It is the last enzyme in the respiratory electron transport chain of cells located in the membrane. It receives an electron from each of 4 cytochrome C molecules and transfers them to 1 oxygen molecule and 4 protons, producing 2 molecules of water. In addition to binding the 4 protons from the inner aqueous phase, it transports another 4 protons across the membrane, increasing the transmembrane difference of proton electrochemical potential, which the ATP synthase then uses to synthesize ATP. The enzyme cytochrome C oxidase, or complex 4, is a large transmembrane protein complex found in bacteria, archaea, and mitochondria of eukaryotes. In fact, oxidase test is also called cytochrome C oxidase test, and this test is actually used to check the cytochrome C oxidase in microbiotics. But how does it work? The oxidase test uses discs impregnated with an EDOX indicator reagent such as NNNN, tetramethylphenyldiamine, TMPD, or NN-dimethylphenyldiamine, DMPD. The reagent is a dark blue to maroon color when oxidized and colorless when reduced. Oxidase-positive bacteria possess cytochrome oxidase. These both catalyze the transport of electrons from donor compounds like NADH to electron acceptors, usually oxygen. The test reagent acts as an artificial electron donor for the enzyme oxidase. The oxidized reagent forms the colored compound indophenol blue. For example, if we use TMPD as reagent, when cytochrome C oxidase present, it oxidizes the TMPD to indophenols, a purple or dark blue color end product. The cytochrome system is usually only present in aerobic organisms that are capable of using oxygen as the terminal electron acceptor. Here we take filter paper spot method as an example. The procedure of the test is: add a few drops of the solution to filter paper, remove a colony from the blood agar plate using a sterile toothpick, then smear it onto the most-stinned filter paper. Then you can check the color. A positive reaction is indicated by an intense deep purple hue, appearing within 5 to 10 seconds, a delayed positive reaction by coloration in 10 to 60 seconds, and a negative reaction by absence of coloration or by coloration later than 60 seconds. Oxidase test can also be done with plate, swab, or test tube. The result is also defined by color. Precautions and limitations. Timing is critical to accurate testing. Use fresh reagents, no older than 1 week. Older reagents can auto-oxidize, thus giving erroneous results. Do not use if the reagent or filter paper is purple. Do not test organisms growing on media that contain glucose or dyes, e.g. McConkey agar or EMB agar. Do not use nickel-base alloy wires containing chromium and iron, nichrome, to pick the colony and make a smear, as this may give false positive results. Bacteria grown on media containing dyes may give aberrant results. Older cultures are less metabolically active, so may give false negative results within the mentioned observation time. The oxidase test must be performed from 5% sheep blood agar or another medium without fermentable sugar. Fermentation of carbohydrates results in acidification of the medium, e.g. lactose in McConkey agar or sucrose in TCBS, and a false negative oxidase test may result if the surrounding pH is below 5.1. Subinoculation on nutrient agar is required before the oxidase test can be performed. During the identification of suspected Vibrio cholera isolate, it is not possible to perform an oxidase test directly from a TCBS culture because the acid produced by the sucrose fermenting colonies will inhibit the oxidase reaction. Oxidase test is most helpful in screening colonies suspected of being a member of the Enterobacteriaceae family. All the members of the Enterobacteriaceae family, including E. coli, are oxidase negative. To avoid misidentification, perform an oxidase test on all gram-negative rods. Oxidase test is especially important in separating Aeromonas from Enterobacteriaceae. Note, if you see swarming colonies in a culture media, do not perform oxidase test, as its unique characteristics of Proteus spp, which are oxidase negative. Oxidase test is used as a major characteristic for the identification of gram-negative rods that are not in the Enterobacteriaceae family. Colonies suspected of belonging to other genera, Aeromonas, Pseudomonas, Neisseria, Campylobacter, and Pastorella, are oxidase positive. Gram-negative diplococci give a positive reaction. All members of the genus Neisseria are oxidase positive. Moraxella spp, that are either gram-negative diplococci or coccobacilli, are also oxidase positive.